Okay, why don't we go ahead and get started? Welcome, everyone. It's always good to see a decent turnout in between terms. We've actually had a number of these, and I've already apologized to the fellows of the ones that I'm going to have to miss due to some travel commitments. But anyways, today is Julian Egger's doctoral symposium, which probably, as most of you know, is kind of the first step towards the dissertation in terms of giving a background talk, kind of preparing for the research that you're thinking about doing, and then that's followed by the eventually the proposal defense, and then at the end the defense of the dissertation. Anyways, a little bit about Julian. He received a bachelor's degree in bioinformatics with a minor in computer science from University of Nebraska, Omaha, and has worked a few jobs in between coming here as a bioinformatics programmer at the Omaha Denry-Dorley Zoo Genetics Department, looking at evolutionary genomics of species from Madagascar. Also spent some time as a bioinformatics software engineer for Thermo Fisher, coming here has developed a research interest in the study of genomics using RNA-Seq with an emphasis on alternative splicing and cellular differentiation and disease. So he will present his symposium today.
All right, thank you, Bill, and thank you, everybody, for coming to my symposium. So my symposium is Differential Splicing Analysis with RNA Sequencing, Current Applications, Approaches, and Limitations. Just a brief overview about what my talk will entail. I'm just going to give a brief background on alternative and differential splicing, what it is. And then I'm going to do basically a top-level overview and comparison of the different methods used for differential splicing and some of their strengths and weaknesses. And then I'll also go into some limitations with the current methods and also some open areas for studying differential splicing with RNA sequencing. And then after that, I'll leave it open for questions. So starting out with the central dogma of biology, it was a term coined by Francis Crick in 1958 to basically describe the process in which the DNA for a particular gene can be made into an actual protein product. And so the central dogma states, of course, proteins being the key component of cells, or the key building blocks of cells, the central dogma states that DNA is first transcribed in RNA, and then RNA is then translated into protein. So why do we have this intermediate step? Well, first off, so transcription actually occurs within the nucleus of the cell, which is where DNA is contained in a tightly packed fashion. After transcription occurs, the translation process from RNA actually happens after the RNA is then taken out from the nucleus. And this happens to ensure that the DNA that we need to stay protected and prevent from any sort of DNA damage can stay protected within the nucleus, and that any sort of machinery for the transcription translation or the translation process of the RNA can all happen outside the cell, and the DNA can be protected from any of this. This also happens, though, for an efficiency purpose. One key to cellular regulation is not just what genes are expressed to make what proteins, but also how much of that gene is expressed to make a certain amount of proteins. And so by doing this, we have an improved efficiency where we can make multiple RNA templates from a single gene, and then from there we can make multiple protein products from that same gene. So if we look at this a little bit closer, what actually is going on that the central dogma doesn't show is that when a gene is transcribed, we actually first have a pre-mRNA product. And then this protein complex, known as the spliceosome, comes in, and it recognizes these different sites in our pre-mRNA product, known as exons and introns, and then splices these sites out, taking the exon sequences and forming them together to produce a full mRNA, removing the intron sequence away, and then this final mRNA product is what's used to make the actual protein. Looking at this a little bit closer of what's actually going on here, essentially what we have is we have our pre-mRNA sequence, from our RNA, which was transcribed from our DNA, where we have all the same bases except for all of our thymine is now replaced with uracils, and the spliceosome protein complex is able to go by and recognize what are known as splice sites, and it can recognize which sequence or regions of sequence should we use to actually include in our final mRNA product, and which other sequences, our introns, are we going to use to actually remove and not include in our final mRNA products. Even more so than this, not only do we have certain segments that we're going to use for a final mRNA product, we can actually pick and choose which segments that we're going to include in our mRNA product, and from there we can actually produce multiple unique transcripts from a single gene. And so this happens all the time. In fact, for multi-exon human genes, over 90% of those type of genes actually undergo alternative splicing. And the end game being here is not only do we have multiple transcripts, we can also potentially produce multiple proteins, each with their own unique, possibly different functions or antagonistic functionality. So there's different ways in which we can actually do this. So in terms of splicing, we could just include, like if we look at individual regions of alternative splice transcripts, we can include constitutive exons and use all the exons. Or what we could do is we could use what's called a cassette exon, or more simply put, exon skipping, where we can actually just, either include or exclude this exon to produce two different transcripts based on, including or excluding this exon. We can also do other things where we could have alternative splice sites, where we can actually incorporate different regions of the exons that we're going to use. We can, have that, for this case, this 5' splice site, we can either include or exclude this particular region of the exon and then concatenate it with this next exon, or concatenate this exon from this and use, different regions of this exon. We can even, in some cases, we can also do what's called intron retention, where we actually continue to include the entire intronic region, or at least some of the intronic region. Or we could do something a little bit more complicated, where we have mutual exclusivity, where we might include one or the other exon. So a few significant examples of this. For instance, there's what's known as tissue-specific alternative splicing, where we might have the same gene expressed in two tissue types, but we might have only one of the alternative splice transcripts expressed in one tissue type, and that same alternative splice transcript isn't expressed in another tissue type, and then the resulting proteins in these two tissues are completely different, and then they'll have different functional domains of that protein, which can then interfere and create tissue-specific rewiring of protein interactions. In the disease case, we can also have what's known as aberrant splicing, or missplicing, essentially. So like a good example of this is where we might have a mutation in one of our exons, which disrupts the spliceosome machinery, and that leads to an inappropriate exon skipping event, leading to a potential oncogene. So differential splicing, this is a little bit different than alternative splicing. And so first off, what I'm talking about here, most of us are at least familiar with differential gene expression, where we're looking for a significant change in expression of a single gene between two different groups. And so when we look at gene expression, we're actually, what we're doing is we're quantifying the total mRNA that's transcribed from a single gene. So we might have, a single gene X, and we want to see what's the change in expression between, maybe a cancer and a non-cancer patient, or cancer patients and non-cancer patients. Based on what I just explained about alternative splicing, it's not that we just have one single mRNA. We actually have multiple mRNAs from alternative splicing. So we can have multiple pre-mRNAs that are alternatively spliced. And so actually what we're looking at, when we're looking at gene expression, we're not just looking at the expression of one transcript. We're looking at the total expression of all isoforms from all our alternately spliced transcripts. So we can see here, when we're comparing these two groups at the gene expression level, this is actually a summation of all the different isoforms that make up that gene. And so looking at this all together, here's another example where we have a single gene, we have two alternately spliced transcripts, and if we look at the total expression, we have a slight change. We have a downregulation in our cancer case, but we don't actually necessarily have a significant change in differential expression. What we do have is a significant change in our different isoforms expression, and then even more so than that, we have what's known as differential isoform usage, where it's not just the total change in expression of the different isoforms, it's the change in the relative usage of these isoforms expression to the total expression of the gene. And so that's what I'm going to be talking about going forward here, is what differential splicing is. So differential splicing is a difference in the expression of all alternately spliced transcripts relative to the total expression of the gene between two or more conditions. So again, looking at this case, we have two isoforms. We might have the same expression at the gene level, but what we do have is a significant change in the proportion of expression between the isoforms that make up that gene. And just to avoid confusion going further, there's a lot of synonyms when it comes to differential splicing. I'll mostly be referring to it as differential splicing, but you've already heard me at this case, refer to it as differential isoform usage, isoform switching, differential transcription, etc. And then going back to a few examples of actual differential splicing in biology and disease, if we look at our tissue example again, it's not so much that we might just have, one transcript expressed in one tissue and the other and another transcript only expressed in another tissue. What we'll probably have is actually we have both transcripts are actually expressed in both tissues, but what we have is a differential usage or a differential relative usage of those, where one transcript is completely expressed a lot more than the other transcript in one tissue versus the other tissue. And then going back to another cancer case, there's a really well-studied case of this for the BCLX gene, where the two alternately spliced transcripts produce proteins with antagonistic functionalities, where one is pro-apoptotic and the other one is anti-apoptotic. And as you can imagine, in the cancer cases, this anti-apoptotic has a much more dominant expression than the non-apoptotic version. And probably one of the most significant cases of differential splicing that contributes to biology is in cellular differentiation. A particular cellular differentiation program known as epithelial to mesenchymal transition is basically a developmental program where we have these tightly compact surface barrier cells that are immobile, and they undergo a differentiation program where they slowly change into these mesenchymal cells that have a lot more motility and a lot more are a lot more loosely organized together. And how this transition occurs is, well, it's driven by a distinct change in overall gene expression. So hundreds of genes are either downregulated or upregulated, so these cells these epithelial cells can then differentiate into these mesenchymal cells. More so than that, though, we also have these changes further driven by expression of splicing events, where we have expression of, we might have alternately spliced transcripts from the same gene that are both expressed in both cell types, but we have an increase of certain splicing variants in one cell type and a decrease. And this change occurs that helps drive forward this change in differentiation. And so this is really a good way, I think, to kind of put together what differential splicing is and how it kind of contributes to overall gene expression, where, we have our change in gene expression profile. So looking at, this is a plot showing differential expression where we have our epithelial cells and our mesenchymal cells. We can see which genes are differentially spliced. All the black dots, all differentially expressed. All the black dots represent differentially expressed genes. And then all the dark gray represents differentially spliced genes or genes that are both differentially spliced and differentially expressed. So we can see all of our differentially expressed genes. We also see in the middle here, these genes, the ones that are dark gray, aren't necessarily differentially expressed, but they're differentially spliced. And then we also have cases where this dark gray on the outside kind of takes up both cases, where it's both differentially expressed and both differentially spliced. And so we can kind of look at it as where the differential expression kind of tunes this differentiation program, and then the differential splicing of these genes sort of fine-tunes this program. So it kind of further drives the phenotype of these two cells. So how can we go about studying differential splicing? Well, one way we can do this is by utilizing RNA sequencing. And so the goal of RNA sequencing is to measure the dynamic transcriptome of cells, basically. And so I say dynamic in the sense of the transcriptome. It's not only does it represent all the RNA, but the transcriptome itself is dynamic in that we have different expression of that RNA or different amounts of that RNA. So when we look at RNA sequencing and we look at the transcriptome, we're studying the transcriptome both in a qualitative fashion, where we're looking at the actual RNA sequences that are present within it. We're looking at the alternative splice transcripts. We're also looking at the expression of that RNA and the expression of those alternately spliced transcripts. Just a really brief overview and basic workflow of how RNA sequencing works. Essentially what we do is if we want to look at the transcriptomes between two different samples or two different groups, for instance, two different cell types, what we'll do is we'll isolate all the mRNA from both those cell types. Due to the nature of the sequencing, we'll actually create cDNA fragments just because it's more stable than RNA. And then we'll fragment all that cDNA up to kind of different sizes with different lengths. And what we'll do is we'll sequence all these cDNA fragments in a massively parallel fashion, producing hundreds of millions of these short reads. So basically, our entire transcriptome from each one of these cells is contained in this big kind of globular mess of hundreds of millions of short reads that make up our transcriptome. So what do we do from there? We got to piece it all back together. So here are all of our reads, where all of our transcriptome is, and basically we have to sort of make out everything that's there and kind of piece it all back together. So how we do that, we can align it to a reference genome, which is a sequential representation of our genome. And we can align these sequences to identify where they're at, but we can also utilize what's known as transcriptome annotations. And so these transcriptome annotations basically represent the structures of our genes in the sense of transcripts and the sense of exons, where, for instance, this is a single gene, and these are two alternately spliced transcripts from that gene, and these represent the positions of the exons relative to the actual reference genome. And then so when we align our reads down onto our genome, we can actually make out the structures, and then more importantly, we can also quantify the different transcripts that we have here from our RNA sequence reads, utilizing the annotations aligned to the reference genome. And a few things you'll kind of notice right away, and I'll talk about this more as we go, is that certain reads, first off, is that when we align to the genome represents where our genes would be before splicing. So, but you have to remember, when we isolated this mRNA, this is actually from fully mature process mRNA post-splicing. So when we go back and align it, a splice-aware aligner will actually split the reads at where the splice junctions were. And so based on, if we have unique splice junctions between the transcripts, we can actually disambiguate which reads belong to which transcript. But you'll see right away we also have issues where we have certain reads that if they align in the same position that are shared between, where an exon is shared between two or more transcripts, we have this ambiguous case where we're not really sure which is the transcript of origin for these individual reads. Another thing to keep in mind is these transcriptome annotations themselves. Depending on what organism we're studying, a lot of times these transcriptome annotations are actually fairly incomplete. Even for really well-studied organisms like humans and mice, we still have incomplete annotations for certain genes. And even more so than that, we might also have cases of aberrant splicing, where we might have some sort of novel splice site occur that isn't contained within our transcriptome annotations. So when it comes to actually analyzing differential splicing, there's actually different methods we can take. And so there's sort of these three general methods. And even though biologically we might be looking at the same thing, we can actually go in and look at it a different way. And from there, we can actually ask different questions about our data. So what I'm going to do, this isn't meant to be a huge laundry list about different methods, but what I'm going to do is kind of describe the different approaches that these take and kind of explain how they're different and how they're actually able to ask different questions. And in doing so, I'm going to use basically a use case of an example piece of software. And these three software are pretty much the most utilized software for each one of these approaches. So the first one I'm going to talk about is what's known as the isoform resolution approach. And this is probably the most intuitive approach for looking at differential splicing based on how I explained alternative splicing works. So basically what we're doing is we're going to look for the differences in the complete isoform proportions or the expression of those isoforms between different samples. And so the software in focus here is the software known as CUFDIF. And the experimental approach that CUFDIF takes is that it tries to assemble all full-length isoforms from our data, estimate the expression of all the transcripts, and then test for relative differences between two or more samples. So CUFDIF is actually part of an RNA-Seq suite called Tuxedo, where this suite is basically used for assembling, quantifying, and testing different genes and transcripts for differential expression and differential splicing. The module cufflinks is actually what does the assembly and the expression estimation, and then CUFDIF is what actually does the testing for differential expression and differential splicing. And so what cufflinks does, it utilizes all of our reads to our reference genome. It also has the option of utilizing transcriptome annotations, but it's not required. And what it tries to do is it tries to find all these mutually compatible fragments to try to disambiguate different transcripts from the data. And so it does this by finding the minimal amount of transcripts that best explains the aligned reads. It uses what's known as a weighted bipartite graph, and then it tries to assemble the transcripts based on the data. It then has to use sort of an estimation approach to actually figure out the actual expression of our individual transcripts. So it has to model this, and you can see right away we have the same issue that I talked about earlier, where we have certain splice junctions that are unique to certain transcripts, but if we have reads that align to ambiguous regions, we're not able to actually identify the transcript of origin. After quantifying, or after identifying and assembling all of our transcripts and estimating the abundance of all those transcripts, we can then do the actual differential splicing test. So this is where CuffDiff comes in. It looks for a change in isoform abundance using a one-sided t-test of a distance metric. This distance metric is known as the Jensen-Shannon divergence distance metric, where we can test basically use a distance metric to test the difference in the relative abundance of isoforms between two or more samples. So isoform resolution models. So kind of what are they good for, and what, why are they beneficial? Well, the most obvious reason is that we actually can resolve and look at full-length isoforms, which is kind of the intuitive thing that we want to do. So we could, in this case, we could look for isoforms that underwent significant differential splicing or significant isoform switching. We could identify potential biomarkers if, we find a gene that had significant isoform switching and also showed a significant stratification, for instance, like in a survival analysis. We also have the ability where we can actually look at look at predictions about the proteins that those different isoforms actually encode. So if we have differential splicing of a particular isoform, we can look at what domains were affected by the differential splicing and what how that actually interferes with things at a network level or at a protein level, and how that might interfere with protein networks. But again, there's this limitation that we have with these isoform resolution models, where we have this ambiguity with reads aligning the same position. So isoform resolution models, although they're nice and we can do certain things, there's a lot of complexity that comes with it. So there's kind of different approaches we can take to try to resolve some of this complexity that we have from trying to actually assemble full-length transcripts and try to estimate their expression. What we can do is a more isolated approach, known as an exon usage approach, where we try to actually look at differences in exon-specific expression. So exon usage approach, in this specific piece of software called DEX-seq, its experimental approach is to actually focus on differential exon usage. And like I mentioned before, it tries to avoid that complexity that we have with trying to actually resolve full-length transcripts and trying to estimate their expression by actually narrowing it down and look at the individual exons for all the different alternately spliced transcripts. So it equates differential splicing with basically differential exon usage or differential expression at the exon level. So how this works, the exon usage model by DEX-seq, it utilizes the transcriptome annotations provided by the user, and what it does is essentially flattens all of our exon models into sort of like this single unit, and then any sort of overlap that we might have with overlapping exons, and then splits it into non-overlapping exonic bins. It then quantifies those exonic bins just like you would quantify maybe like a single gene separately for each individual bin based on our read densities. And then when we go to test for differential splicing, what it does is it uses what's similar to actually a differential expression approach. It uses a generalized linear model to test for differential exon usage or differential expression at the exon level. Only at this time, our generalized linear model now accounts for the total expression of all the exons within the gene. And then we can look for basically significant differential expression of a certain exon and look for significant exons that underwent differential splicing based on like an FDR cutoff. And we can kind of utilize this in a different way. Although we didn't resolve our our full-length transcripts, we might actually have a more accurate approach because we didn't have to do that abundance estimation at the transcript level, where we can actually just utilize the results at the gene level, where we can say, okay, we know that this gene underwent some form of differential splicing, and we could therefore use that gene as a potential biomarker, or we could even just use that exon, and that differentially spliced exon could actually be used as a particular biomarker. So a splicing event approach kind of extends upon that exon level approach, where, again, we want to avoid that complexity of trying to resolve transcripts, but rather than just looking at the individual exons, we basically want to try to add some sort of biological relevant information to it, where we're going to actually look at different splicing events that we could actually have. So the software I'm looking at is the software called RMATs, where it focuses on localized splicing events. So through all of our data, it's going to look for these different kind of splicing patterns. It's going to equate differential splicing with basically a differential inclusion of these different events. For instance, whether or not do we have this skipped exon or do we not have this skipped exon. And like I mentioned before, it extends upon this exon-based approach by adding biologically relevant information. So how it does this, so again, we're looking at our transcriptome annotations. What it's going to do is going to identify different splicing event patterns that are found between all of our transcripts. So in this case, we have 4 transcripts, and we have an exon skipping event where three of our transcripts include the exon, and another transcript doesn't include an exon. And what we're going to do is we're going to flatten those structures down into a single unit, and now we can actually look at the inclusion or exclusion and of basically just 2 cases, just the inclusion of the exon or exclusion of the exon, and we can quantify. No longer do we have to quantify using all these individual transcripts. Everything can be quantified right here on this level. And we quantified that using what's known as percent spliced in, was essentially of the total expression of these 2 cases, what's the percentage of the transcript being expressed that, not necessarily the transcript, but what's being expressed on the exon, for the exon inclusion versus the exon skipping. So we have the total expression of these guys, and then, like, for instance, maybe 0.7 of the total expression, maybe 3/4 of the expression is coming from the exon inclusion event. When it comes to actually testing for differential splicing, what it'll do is it'll determine the change in inclusion between 2 or more samples, and then use a likelihood ratio test to determine the p-value for a change of percent spliced in exceeding some sort of defined threshold. For instance, we could look for significant change in splicing that's greater than maybe 5% or 10%. And so what this might look at, so we kind of localized our view. We're looking at a single gene across for 2 samples with 3 replicates for each sample. And we're testing the inclusion or exclusion of this particular splicing event. And we can see here between these 2 samples, we have a change of percent spliced in about 10%, where relative to the overall expression, this one has a much, in this sample, we have a much lesser extent of contribution to this included exon than in the other sample. So how are these useful? We can use these a lot of different ways. They're really nice. They might be a little bit more accurate in the sense that we're not looking at full transcript complexity, but then we can actually use these splicing events for, again, for particular biomarkers. For instance, we could look at different splicing events across different cancer types and maybe identify significant biomarkers in the form of splicing events. So what I really want to get into is some of the limitations and then open areas for differential splicing analysis. And so one of the first kind of gap in an open area for differential splicing is what's known as overlapping transcriptome features. So what do I mean by overlapping transcriptome features? So basically there are features, we can look at this in the sense of our annotations, features that basically overlap in either some or partial manner. And again, these features, what we're really looking at is all these individual exons, but kind of recall this transcriptome feature hierarchy where we can have genes, for instance, this is a single gene that can have multiple transcripts, and then all these multiple transcripts are made up of individual exons. So for the most part, we're looking, when I say overlapping transcriptome feature, we'll be just looking at these individual exons relative to their position along the genome. So overlapping genes. So keep in mind, DNA is double-stranded. So we can actually code for proteins using both strands of DNA. So this leads to the potential where we might have overlapping genes going in opposite strands that are actually now utilizing kind of the same regions of the genome for the same different exon, but on opposite strands. However, we can also have cases where we can have overlapping genes going on the same strand, where 2 different genes can actually, on the same strand, utilize some of the same exact exons to make 2 different genes. And this actually happens quite a bit. So this is a study looking at a set of annotations, these GenCode version 19 annotations, to see what was the extent of overlapping at the gene level. And one thing to kind of keep in mind is that this is including only the coding regions, so it pulls out only the protein coding exon regions from all the genes and looks at the degree of overlap across every chromosome. So we can see we have a much greater amount of overlap on the opposite strand versus the same strand. For opposite strand, we have anywhere between 10 to 30% of our genes are overlapping on a single chromosome. And even though it's not as much, we still have a significant amount of overlapping genes that are found on the same strand. So anywhere between 5 to 15% of genes on chromosomes are overlapping on the same strand. This is just showing the distribution based on how much of the exon protein coding regions of a gene overlap and what's the degree of genes that actually have this much overlap for both same strand and opposite strand overlapping genes. And then keep in mind, like I talked about earlier, we can also have overlapping features just within genes, so due to alternative splicing, where a single gene can have multiple transcripts sharing all or some of certain exons for those transcripts. And now if we include overlapping exons from alternately spliced transcripts and not just overlapping genes, we can see that we have upwards, especially in humans, we have upwards of hundreds of thousands of exons that are actually going to be overlapping. So this presents a lot of issues for analyzing differential splicing. So either way, whether they're alternatively spliced genes sharing exons or they're overlapping genes on the same or the opposite strand, they present problems. So how do we handle this? So first off, with overlapping features on the opposite strand, luckily there is something we can do now, is what we can use what's known as strain-specific library prep, where there's a lot of ways to do this, but essentially what we're doing is we're able to basically tag our reads into saying which strand they were actually sequenced from. So in the case of where we do have overlapping, we can kind of resolve that ambiguity issue and actually see, okay, this is on one strand. We know all these reads had to come from that strand, and all these reads had to come from another strand. So when we have an overlapping case, right away, even though they align to the same genomic position, to like the same exon structures essentially, we can actually disambiguate that and know which reads belong to which strand and can kind of deal with this overlapping gene issue. However, we still have the same strand issue, which is kind of left, where we can have overlapping genes, overlapping features from the same gene or overlapping features from different genes, and they all present issues to these different approaches for differential splicing. So for the isoform-based approach for overlapping genes, remember we have this ambiguous issue where we can't assign ambiguous reads to the individual transcripts, and so kind of how cufflinks has to do this is it needs to assign these different reads to the right isoform of origin. So it uses a maximum likelihood estimation to try to determine, okay, what's the most probable isoform that one of these reads could have belonged to. So by doing this, we get an estimation with a certain bit of uncertainty, and now so all of our expression for each one of our transcripts that we're looking at is now based on a probability with a given sense of uncertainty, and it's those uncertainty estimates that are used in the actual testing for differential splicing between two conditions. And so a key problem of that to keep in mind is that as we increase the number of transcripts that we might have and we increase the amount of exons that are shared or partially shared, we decrease our certainty in the estimation of the expression of those different transcripts. So when we go to overlapping genes, this amplifies the issue. And the cufflinks approach to do that, so in the case where we have two overlapping genes with two alternately spliced transcripts for each gene, what cufflinks does, it actually groups all the overlapping genes into a single group, and then it goes and it estimates the relative abundance of each transcript from each gene within that group, and then does the test that way between the two conditions. And this leads to obviously a lot of issues. We have that ambiguity issue of aligned reads, but we're also unable to distinguish splicing at the gene level. So for instance, if we detect splicing, we don't actually know which gene was actually differentially spliced. And again, like I mentioned before, by doing this, we increase the number of transcripts, we increase the probability of having exons overlap, which decreases our certainty for our abundance estimations, and we lose power to detect significant changes in transcript abundance. So the exon-based approach for overlapping genes, if we recall the approaches for alternatively spliced genes, where we take our annotations and we kind of compress this down into the single structure, DEXseq takes maybe one of two approaches to look at overlapping genes. So one way we can do this is the same thing we did for the alternatively spliced genes or the alternatively spliced transcripts. We can compress all of this together now, where we have all the exons from all the transcripts from all the gene into basically a single unit, where all the overlapping exons are split into individual exonic bins. Or what we could do is a separate test where we actually just take any overlapping features and completely remove it for our test. So if we test it this way, we'll test the genes separately, but we'll only be doing on the exons that weren't shared. So we're not going to be doing any sort of test on, for instance, these exons. So this presents a lot of issues. So obviously we have that issue of lost exons, which, like I showed you earlier, in the case of humans, we could have hundreds of thousands of exons that are overlapped that could be lost by this approach. Again, we have, just like the isoform resolution issue, we have ambiguity at the gene level, so we don't know which genes are actually undergoing differential splicing if we're testing them as a single group. And if we do this, and kind of another issue is we can actually mistake differential gene expression for differential splicing. For instance, if we tested this particular exon and this gene was actually differentially spliced, this exon, there's certain cases where this exon would actually be picked up as differential exon usage when it's actually differential gene expression. So the splicing event approach for overlapping genes is a little bit different. So we have our two genes overlapping. What the event-based approach does, just like before, it goes and identifies all the different splicing events from the transcriptome annotations. And then all the events are annotated separately for each event for each gene. And so there's no regard to overlap. And then so each test for each event is going to be done separately for each gene. So we're not grouping genes together like we did with some of the other methods. The problem comes, though, for, again, the ambiguous read issue, where we might have inaccurate estimations, because what RMATs will do is any sort of ambiguous reads that could go to either gene, it's just going to count the expression of, it's going to count this read towards the expression of both genes. So that, could potentially lead to, inaccurate estimations of our actual percent spliced in, which would therefore lead to inaccurate changes in percent spliced in or inaccurate differential splicing. But the event-based approach is actually is actually a pretty good approach to take for differential splicing. It's actually probably the most used method, I would say. And again, one of the reasons for this is that it avoids that complexity of trying to resolve transcripts and trying to estimate different abundance of transcripts. But also, unlike the exon usage case, it actually tries to incorporate biologically relevant information. And kind of why it's so good is that we can now compress all these transcripts into a single unit, and by quantifying this just on the single unit and not on these different transcripts, we can actually increase our power to actually detect differential splicing, or at least differential inclusion of this event. And it's very useful. Again, we can use, just like a skipped exon, for instance, as a significant biomarker if we want. Again, like using a survival analysis as an example, we can just, instead of trying to look at all the transcripts, for instance, for the STAT5A gene, we can actually just look at this skipped exon or not. However, the problems that come with event-based models are complex alternative splicing events, where event-based models like RMATs are actually only looking at splicing patterns that kind of fall into one of these 5 categories. It misses any sort of pattern that we could have, and there's a lot of them, that aren't, just simple exon skipping or just a simple differentiated splice site or a mutual exclusive exon. It misses any of those. And additionally, it also only looks, all the events that it looks at are strictly binary. It's always, inclusion or exclusion of like a skipped exon. It's always, one site or the other site, or this exon or that exon. It can't handle anything that might have maybe 3 or more possibilities for one particular splicing event. So the limitation of event-based models is complex splicing events. And we actually have a lot of this. So if we look at another study looking at trying to annotate different splicing patterns through our annotations, we can see for 2 really well-studied organisms, we have almost 20% of our splicing patterns actually don't fall into one of these 5 categories. So what is complex splicing besides just not falling into one of these? Well, kind of a few examples. There's different ways we could look at this. One could be compound events, where we're actually just using multiple simple splicing events, but all in sort of one fashion. Or we could have, for instance, an alternative splice site compounded with a skipping event. We could also have multiple skipped exons. So event-based methods usually only are able to detect, one exon being skipped. We actually have cases where we might have like a cluster of exons that can be skipped. A really good example of this is this BARD1 gene, which actually produces multiple alternative splice transcripts, where the alternative splice transcripts undergo extensive exon cluster skipping. And these different transcripts have been found to be differentially spliced in different cancer types. And these the different proteins that are actually translated from these different transcripts all have unique interactions with the BRCA1 gene in breast cancer. So yeah, the event-based model would be missing all these clusters. And then, like I kind of mentioned before, non-binary events, where it's not just inclusion or exclusion of one event. We could have cases like in this example, this CAMP-K2G gene. It's a non-binary case where we can actually use any combination of these group of exons right here. And looking at here, this is RT-qPCR showing the expression of these different combinations across different mouse tissues, where we actually have preferential expression of these different splicing patterns across different tissues. And then finally, so this is my last kind of open area that I want to talk about, which is kind of more of kind of falls into an umbrella case of just trying to improve the annotation and quantification of alternative splicing so we can use them for differential splicing tests. And this particular thing I'm going to talk about is just the idea of utilizing both novel and ensemble approaches to try to better annotate and better quantify the different splicing events so we can use them for differential splicing tests. So looking again at event and exon-based approaches, again, they're really great methods. They resolve that complexity issue, but the thing about them is that they're highly dependent on the transcriptome annotations. They're very limited if we're doing this type of test on, for instance, a particular organism that doesn't have very complete transcriptome annotations, or in the case where we have some sort of novel splicing from, for instance, from aberrant splicing, they're limited in that they can't actually detect that. So there's one kind of idea is the use of alternative splicing graphs. So alternative splicing graphs have been around for a while as a way to sort of identify different splicing patterns in different genomes. And one thing we can do by using alternative splicing graphs is we can take our annotations and basically build a graph representing all of our variant splicing patterns that we could have from all of our different transcripts. So we take the annotations and then build this graph representing all the possible variants. And so we can use this, for instance, if we want to build a event-based method, we'd actually, instead of trying to find all of our events from annotations, we could first build our graph and then from there actually try to identify events. Well, okay, how does this help us? What we can also do is we can also not just utilize our annotations, but similar to an isoform resolution approach, we can also utilize our data. We can utilize our aligned reads to also add to our alternative splicing graph. And in this case, now we'd be able to detect novel splicing events. And then going a step further, we could then possibly annotate and quantify complex splicing events that don't fall into one of those 5 basic splicing patterns that the typical event-based methods would actually take. So there I'll kind of conclude and summarize just that differential splicing is an often underutilized analysis from RNA-Seq studies. Although it's something I think that is worthwhile, annotating and quantifying alternative splicing is still inherently difficult due to problems that we have with RNA-Seq. It's a much more complicated process than just looking at expression at the gene level. There's multiple approaches, all of which have their different advantages and all of which are actually asking different questions that could be helpful in different ways. However, limitations still exist with all these different methods. Like I mentioned, a few overlapping features and complex splicing being really good examples of that. And then also that I think that using both novel and ensemble approaches for finding and testing differential splicing may actually improve the robustness of analysis. So with that, I just want to thank again OHSU, the DMI student, faculty, and staff, all the BCB fellows who were helping me put together this talk as I was doing this. Definitely want to thank my symposium committee who are here today, and also acknowledge my NLM training grant. And with that, here are my references, and I'll leave it open for questions. Uh-oh. ECC. Oh, that's just like a bioinformatics. Like, you say all the EC. So say that again. You said EC-Seq.
You were saying that EC-Seq was a technique for strand-specific library preparation.
Oh no, you might have been referring to the.
Was I in the wrong?
That was just a citation for that where I got the figure for.
I see.
EC-Seq. Yeah, that's just like a bioinformatics resource website explaining bioinformatics. Yeah.
Then my question is, what databases exist that have strand-specific sequencing data that one could use?
Oh well, that's just part of the RNA sequencing. So to answer your question, databases that would just be like short, like SRA, the short read archive.
Well, yeah, is it a technique for library preparation?
Yeah, it's just a technique for library preparation.
But so everyone does it? Is that what you're saying?
Not everybody. It's before it used to only be unstranded, but now a more like modern improvement to RNA sequencing is that we can do stranded prep. So if you look at, if you get some RNA sequencing data, you want to know if is it stranded or unstranded or not. So yeah, the data you've looked at is probably listed as being stranded or unstranded.
The data I look at is already summarized. So I can't really.
Right. It's just like, one thing I didn't talk about to not get too technical, it's just like paired end reads. I didn't, you could have single end reads or paired end reads. We could also have stranded or unstranded reads from our RNA sequencing.
Okay.
I'll go ahead and ask. Actually, I'll make a comment and a question. Actually, there's a few people in this room who are who are about my age. We learned in college the central dogma, and it was so much simpler back then.
Yeah.
Without all that other stuff.
It's still it. Yeah. That's still how it is. It's just if we kind of zoom in, there's actually a lot more.
Well, it seemed that,
yeah.
and we didn't know introns and exons
Right.
and so forth. But my question is, what are some of the applications of this? I was not a expert in genomics. I understand the different methods to kind of figure out how the splicing takes place. What are some of the applications of this that you would use in your dissertation, presumably?
All the way to those slides. But yeah, there's a lot of different applications. Some of my interests might be more for, just discovery, trying to look for, how splicing might affect proteins and protein domains, interactions of proteins. One of the example I kind of like is this EMT program I talked about. This differentiation program is actually pretty strongly theorized to be a big part of metastasis, where these cells, these cells would be kind of where the primary tumor would be, and it's strongly believed that the ability to metastasize is that the cancer cells actually hijack this program. So I think, one way, how much we could take with that is maybe trying to understand exactly how cells are able to metastasize and how they're able to hijack this program, knowing that differential splicing kind of plays a key role in this. Going further to more of the clinical sense, what I would actually like to do is maybe look at differential splicing in metastasis patients. So right now we have a lot of data of both normal tissue and primary cancer tissue. I'd like to see paired data and do an analysis in both normal tissue, primary tissue, and then metastasis tissue to try to look for maybe biomarkers at a splicing level to look for patients that are likely to metastasize or might affect survival or treatment and things like that. Just like, and again, I say something like treatment, like that's a good way to look, if we look at gene expression, we can use gene expression as potential biomarkers for maybe treatment options or survival. Looking at differential splicing is just kind of another layer that we might be missing with RNA sequencing, where we could actually identify biomarkers at the splicing level to kind of complement the gene expression level.
I was just wondering if you could comment on the ensemble method that you were speaking of, if the advantages and limitations of actually using existing annotation with your ensemble method.
Oh, the limitations of using existing annotations? Right. Yeah, absolutely. So, I'll go back to that. So one of the key limitations is that it could be biased towards those. So, if we try to build our graph, from those annotations, we might have splicing that's not actually there, but we're using those annotations to sort of build that structure pre-hand, where maybe that splicing didn't occur, but it's being included even though our reads didn't align to it. So maybe a counter approach would be to build maybe two different options where you could build it from your transcriptome annotations, but also just use it completely de novo and do it on your reads. So for instance, we might be forcing our graph to maybe actually include these exons when we actually don't have that in our data, and we're now building our graph from that particular exon just because it was in our annotations when maybe it actually wasn't expressed. That could be a possibility. Shannon.
So how, and this is kind of a looking forward question, how are you, we've had a tremendous change in technology and what we've been able to do in terms of sensitivity, and so if you think about it from that perspective of as you're working on your proposal and work on your dissertation, there's going to be this moving target about us being able to go after lower abundant transcripts, about being able to actually, like for example, I think the annotation, even our assemblies will update because of longer reads. And so have you thought at all about a way to try and be dynamic in that perspective?
A little bit. I mean, I think one example maybe, first off, so she was talking about longer reads, better annotations. Again, one of the limitations about this, I mentioned with the isoform resolution models or any of this, is that the reads are much shorter than our exon structures. So that's where the ambiguity lies. As reads get longer, that can resolve some of this complexity issue. But right now, with the way the technology is, the long reads are actually, we have a much lower depth, so we lose our confidence, and they're much more error prone. So kind of what she's talking about is as that technology gets better, and we can do this at a cheap, it gets better, it's cheaper, it's more available, that'll actually improve our ability to actually look at differential splicing. In terms of kind of going forward with the technology, a really big example I can think of is single-cell RNA sequencing. So single cell, we can actually look at individual cells, and as opposed to just a bulk set of cells. For single-cell sequencing, that's already, and I'm not a total expert on single-cell sequencing, but that's already difficult as it is just for the gene expression level. So you can imagine it's even more difficult if we actually try to look at an alternatively spliced expression or alternatively spliced transcript expression. But there are people who are doing that, and I think that's something I would be really excited for. But that has a really long way to go. And so, yes, doing that going forward. In order to do that, though, there actually would have to be probably a significant change in the approach in certain ways of how we're actually able to handle all the noise and complexity that comes out of single-cell sequencing. But yeah, absolutely. I think for this type of analysis, as years going forward, as the technology better, I think being able to do this will actually get better as RNA sequencing gets better and as we get better, more complete annotations and databases of our annotations and splicing, things like that. Fleming, yes.
Question. Could you say something about like how alternative splicing is controlled, like in different tissue types?
Oh, sorry. You said one time.
Okay, how alternative splicing is controlled or is regulated?
Is regulated in different tissue types?
Yes.
Well, yeah, essentially. So I kind of brought that example up earlier, where we might have tissue-specific differential splicing, where the expression of the different transcripts could be regulated in different ways. So, for instance, we could have two different tissues where, let's say we have two transcripts. Both transcripts are being expressed in both tissues, but the relative usage of those different transcripts are different. So, for instance, maybe like in brain cells, transcript 1 is expressed, contributes to 80% of the total gene expression, where in another tissue it maybe only contributes to maybe 20% of the gene expression. Does that answer your question? Perhaps. No, it doesn't?
What actually causes it?
Yeah. What actually causes this? Oh, that's a better question. Yeah, so what actually kind of controls the expression of the alternative spliced transcripts are like splicing regulator proteins, which are essentially like transcription factors for alternative splicing, or at least that's how I look at it. So I'm assuming since you're familiar with it, I can explain this. For instance, we might have a transcription factor that regulates the expression of the splicing regulatory proteins. And those splicing regulatory proteins actually bind to cis and trans elements along our sequence that then regulates the expression of the alternative spliced transcripts. So actually one of the things I would really like to look at from a network approach is looking at the correlation of both transcription factors and splicing regulatory factors and their correlation with the actual the quantity of splicing we have. So for instance, we could correlate the total expression of splicing regulatory proteins with the splicing inclusion of the differentially spliced genes, if that makes sense. So yeah, so it's that control by those regulatory proteins could actually are what's going to control the degree of splicing that we have in different tissues. No, that's something I'm interested in. Essentially, it would be like a correlation network, but incorporating multiple elements. Yeah. I said I meant gene. I meant the expression of the gene. Yeah. Mm-hmm. I would look at it more as doing like just a gene correlation network, where you're just where you're doing gene expression, but trying to incorporate maybe both gene expression for instance, the regulatory genes with some sort of, it depends how you quantify it, but with the splicing. So for instance, like percent splicing that I mentioned. Right. So does this regulatory gene that controls splicing, does that expression correlate with the percent inclusion of maybe 2 alternately spliced transcripts?
Actually, do you see people do this type of correlation analysis?
I've seen people recently kind of try it. I think what would be interesting is how they're actually quantifying that. So I mentioned, if we want to sort of correlate expression with splicing, how do we actually correlate that with splicing? What ways we can do that? Dr. Dan Iancu, he has his exon level correlation network. His splicing network is based on like correlations between individual exons. So there's not actually any gene expression integrated within that. It's just the correlation between different exons between different genes. Some other approaches what I've seen people do is maybe they did kind of what I mentioned, where they take the expression of the splicing regulatory genes and correlate that with, for instance, like maybe percent splicing of splicing events, or maybe the actual isoform level expressions. That's another thing I've seen is where they basically build a network that overlays both gene expression and then isoform specific expression within the gene expression. But yeah, I think that's actually something really I'm pretty interested in looking at methods to do that as well. Kristen.
I've kind of mentioned this to you before, but
Yeah.
Is there any like mechanistic like lab studies that have been done that instead of correlating with like quantity, looking at actual like type of splicing, like the idea that like a specific type of splicing factor actually is responsible for a
Certain events.
certain type of event?
Yeah. Probably the best example, I actually kind of showed this when I gave a fellows talk a few months ago, was they were doing a splicing event type of analysis, and there's known splicing regulatory genes, and they looked at the type of events that were present, and then they did a knockout model where if they knocked out different splicing events, or excuse me, if they knocked out the different regulatory genes, they could see which events sort of dropped off. I can't remember if they actually were able to see exactly what, like if there was really some reason between what genes had what events, or if it was kind of just circumstantial, but like, maybe by knocking out this particular gene, we lost maybe 30% of our exon skipping events, and we lost maybe 10% of our alternative 5' splicing events, or so forth. So I definitely think people probably looked at that. It's just like how much detail, could you actually see, right?
Actually, inside the cell, there is so-called splicing machine.
Right.
Yeah, which contain probably over like tens of proteins. So they are controlled by all these proteins.
Right.
Splicing.
Right. Yeah, you mean like the spliceosome machinery?
Yes.
Like right. Mm-hmm.
This will be the last question. As long as it takes.
I'm not proposing any aims or anything like that, or any analysis plans. I'm just giving an overview of my the area I'm interested in, and the background and what I understand to this point of this area. So we'll talk about that later.
Okay, thanks a lot.
Hey, thank you.